Hello, class. So if you're watching this, that means that you have completed the ABO-H and secretor PowerPoint. So now we're going to talk about the RH blood group system, and this should be Chapter 5 in your textbook. Now, the RH blood group system is highly complex. It's polymorphic, meaning that it can have many different forms. And if you see here, it says there can be more than 50 different RH antigens. And that's because it's very polymorphic. When the gene is inherited, it's able to change into different antigens. And it's the second most important blood group system after the ABO blood group. So since the initial discovery of the D antigen in 1939, there's been 50 related antigens that have been assigned to this RH blood group system by the International Society of Blood Transfusion, or ISBT. The genetics, nomenclature, and the antigenic interactions are very unsettled. This unit will concentrate on the most commonly encountered observations or the most commonly encountered antigens, the different problems we see with the RH blood group system, and then the solutions. So the terms D positive and D negative refer only to the presence or absence of the RH antigen big D on the red blood cell. The terms RH pos and RH neg are old terms, however, you will still hear it used in common blood bank lingo today, and also blood products are still labeled as such. So there was an earlier name, Rho, RHO, which is less frequently used. The only time you'll really see that prefix is with Rofalac or Rogam, which is actually a synthetic form of anti-D that's given to pregnant women who are RH negative, so that might be the only time you'll see the RHO form. And then there's 4 additional antigens, well, main antigens that we're going to talk about, and it's big C, little c, big E, little e. And these were named by Fisher just because they were the next letters of the alphabet according to precedent, which was originally set by the naming of the A and B blood groups. The major alleles of those 4 antigens are the big C, little c allelic group, and then the big E, little e. Many variations and combinations of the 5 principal genes and their products, antigens, have been recognized. The RH antigens and the corresponding antibodies that can be produced account for majority of the unexpected antibodies that we encounter in the blood bank. And that's definitely true. RH antibodies are stimulated as a result of transfusion or pregnancy. They are immune. So unlike the ABO antibodies, which are expected or naturally occurring, the only way you can really develop an RH antibody, as well as with the other many blood group systems that we're going to talk about, is through an immune reaction. So you have to have been exposed to a foreign antigen that your body doesn't have on its red cells through either transfusion or pregnancy. That's very important to understand. Okay, so the discovery of the RH blood group system, as with many other blood group systems, followed the investigation of an adverse transfusion reaction, or in this case, a hemolytic disease of the fetus and newborn. So key observations were made by 2 researchers, Levine and Stetson, in 1939. And they noticed that after the delivery of a stillborn fetus, there was an adverse reaction in the mom to blood transfusions that she received then from the father. Now, again, remember, things were not regulated back in 1939, so they just took the blood from the father and gave it to the mom. But what they noticed was that she was having a reaction. So the syndrome in the fetus is now referred to as hemolytic disease of the fetus and newborn, so HDFN, and we'll be discussing each of the blood groups' antibodies and whether or not they are capable of causing HDFN, because again, this is a very serious condition. If the mom has an antibody that can attack the baby cells, that's how we get stillborns. The syndrome complicates pregnancies, and it's been complicating pregnancies for decades originally, and if it goes untreated, it can cause severe jaundice and fetal death, also known as erythroblastosis fetalis. So that is the syndrome associated with HDFN that where babies develop jaundice and eventually die. And so erythroblastosis fetalis, or HDN, hemolytic disease of the newborn, is linked with an anti-Rh antibody. And this was noticed by Levine in 1941. So the Rh system was identified. Officially, though, by Landsteiner and Weiner in 1940. What they actually recognized was that there was an Rh antibody that agglutinated about 85% of human red cells tested, and it was actually non-reactive with 15%. And from that discovery, the population was characterized as Rh positive or Rh negative. So that's actually the percentages associated with our population. 85% of our population are Rh positive and 15% are Rh negative. So it was this antibody that was truly the marker for the anti-Rh antibody. The reactivity paralleled reactivity of sera in women who delivered infants suffering from hemolytic diseases. And then a later antigen was detected by using a rhesus antibody and a human antibody, and it was established to be dissimilar but a system already named. So what they're talking about in this case is that a later experiment that demonstrated another antibody that kind of looked like an Rh antibody, only this time it was made from guinea pigs and rabbits. Was similar but not completely identical to the humans, and this rhesus antibody specificity was actually directed towards another similar red cell antigen, and this antigen was actually known as LW, and it was known as LW in honor of Landsteiner and Weiner. So the name of the Rh blood group system was already established, and then they just associated with this rhesus antibody with the LW antigen system. So it's important to recognize that it is possible to see very similar antibodies, but they're not associated with both the Rh blood group system. One is LW, one is Rh. Now, the clinical significance of the D antigen after the A and B antigen is that it's the most important red blood cell antigen in transfusion practice. So individuals who are lacking the D antigen, so those who are Rh negative, they do not have anti-D. It's not an automatic stimulation again of that antibody. Their red cells lack the antigen. So as long as they're not exposed to a positive D antigen cell through transfusion or pregnancy, they're not going to have the antibody. They're only going to develop it if they are exposed to it, and then their immune system will produce it. But the immunogenicity of creating this antibody is greater than that of all other red blood cell antigens studied. What that means is that it's been reported that over 80% of people who are D negative and who receive a single unit of D positive blood or exposed through pregnancy develop that anti-D so quickly. So it's very immunogenic, meaning the body doesn't take time to produce this. If you're exposed to even a single unit, your body's going to produce this immune response. So testing for D is routinely performed in the blood bank so that when we determine if a patient is D negative, we will definitely transfuse them with D negative blood so that they don't produce this antibody. Okay. So now the genetic pathways that create the Rh gene and the Rh antigen in our body is developed because Rh genes are inherited as co-dominant alleles. So you're going to receive one Rh haplotype from each parent. In rare instances, you might see or find a person who has no Rh antigens on their red blood cell. And that's known as an Rh null phenotype. Again, we've seen this word before, null, not producing the expression of a gene. So there are some cases where a person might not inherit any D or any Rh antigens. So there's a couple of different theories that were thought of to explain the inheritance of the Rh antigen. And the most postulated theory is the Tippett theory, and this is describing the Rh blood group system antigens as being encoded by two closely linked genes. So we have RhD and then RhCE, which are both located on chromosome 1. And this is actually the correct theory. So this truly does describe how our Rh antigens are inherited, but I'm going to talk about two others that were actually originally thought about because they are used more so for the nomenclature or how we describe the phenotypes. So we'll get into that in the next two slides, but this is the actual correct theory. There are two closely linked genes on the same loci on chromosome 1. So RhD determines the D antigen expression, so whether it's present or not present. And there is no such thing as an antithetical little d. It does not exist. You either have the big D or you do not. Sometimes, though, you will see that a small little d notation is used to indicate a D negative gene, but just know that even though we use it in our writing, there is no such gene. So if we are going to see a little d, it does mean that they're big D negative, but again, it doesn't really exist. So then the second gene is Rh big C big E. Now this specific gene determines, or this specific allele determines the big C, the little c, the big E, and the little e antigen specificities. So this gene has 4 different types of alleles. You'll see it's Rh big C big E, Rh big C little e, Rh little c big E, and Rh little c little e. And funny enough, these genes are going to encode the red cell antigens based off of the last 2 letters of each of those alleles. So we have the first gene, we'll have a person who's positive for the big C and the big E antigen. The second one, they're positive for the big C and the little e antigen. The third one, they're positive for the little c and the big E. And then for the fourth one, they're positive for the little c and the little e. Another gene that was identified on chromosome 6 is actually known as the RAG gene, RHAG, and it produces an Rh-associated glycoprotein. So RAG doesn't carry any specific blood group antigens, but it associates with blood group proteins in membranes to form the Rh core complex. So you must have RAG. RAG is a co-expressor, so it must be present in order for you to have the expression of the Rh antigen. So not only do you need to inherit the RhD or the RhC, big C, big E gene, you also need to have this inheritance of the RAG, because if you do not, then you're not going to be able to produce any Rh antigens. The other 2 theories that are discussed are known as Fisher and Race, and then the Winer, or the RHHR. So with Fisher and Race, these 2 researchers believed that the Rh blood group system antigens were inherited as a gene complex or a haplotype that would code for 3 closely linked set of alleles. So they believed that there was a D gene, a big C, and little c gene, and then a big E and a little e gene. So they thought there was 3 closely linked genes where each parent would contribute 1 haplotype or 1 set of these Rh genes. So they might have given, a parent might give you a big D antigen, a little C antigen, and a little E antigen. So that would be 1 haplotype. That's what Fisher and Race thought. With Winer, they actually believed, they postulated that the alleles were only located at 1 gene locus, and that this was responsible for the expression of the Rh blood group system antigens on red cells. They believed that each parent contributed 1 Rh gene, and then the inherited form of the gene could be identical or homozygous, or it could be different and heterozygous from each parent. So they actually believed that there was 8 different alleles to exist at this 1 specific locus on the chromosome. And what Winer did was they associated the Rh gene locus as each of these letters. So you have R superscript 0, this is capital R superscript 0, capital R superscript 1, capital R superscript 2, capital R superscript little z, and then you have a little r prime, and then a little r double prime, and then a little r superscript little y. So these genes would each encode a structure on the red cell antigen, and they each identified either, a big D, a big C, little c. So again, they thought this was just one single gene. And now the Fisher race and the Winer, although they are not the correct theory as far as how genes are inherited, these two systems actually developed the nomenclature. So they were used to describe the Rh system prior to any molecular genetics that were developed. They reflected solely only the serological observations, or only the observations that we found through our testing, and they used that to explain the inheritance patterns based off of the different family studies and the different serologic observations. So because they're actually used interchangeably, both the way that we describe the Rh system, so we need to understand the theories well enough to translate from one to another, because when we actually do get into working with blood screenings and antigen identification as well as antibody identification, you're going to find that they use this nomenclature to describe the different Rh antigens. So it's going to be a little confusing, and I have attached some practice problems, but I'm going to try my best to help you understand it. So with Fisher and Race, again, they suggested that the antigens were determined by 3 pairs of genes, which all occupy a closely linked loci. So each gene complex would carry a D or its absence. Again, there is no such thing as little d, but we will call it little d. Big C or little c, big E or little e. So each gene, except for the little d, again, which is an amorph or doesn't produce a product, would cause production of an antigen. So the order of loci on the gene would appear to be, let's say, big D, big C, big E, but many authors actually prefer to put the C before it because they like to alphabetize it. So you might see it in both different ways. But essentially, they both mean that you have the big D antigen, the big C antigen, and the big E antigen. So again, genes are inherited from your parents, and it's in a linked fashion, and you're again going to inherit one haplotype from each parent. And now a haplotype, again, just means that you're inheriting a group of inherited genes. So you could inherit right there, those 2 examples. This could be from one parent, and this would be called a haplotype, and then this could be another set of genes that are linked together, and that would be another haplotype. So the gene little d is assumed to be present when the big D is absent. So if the D antigen is not inherited, then we're going to have a little d in its place. So again, I said there's 3 loci that carry the Rh genes very closely, that they're linked and that they never separate. So they're passed from generation to generation. So this is an explanation of the Fisher race. It's saying that there's a D gene, this big C little c gene, and then a big E little e gene. Again, when it's inherited through a child, they're all going to be inherited together, and they're going to show on the red blood cell surface. So this is a primary linked set of antigens. So below an offspring of, here's a haplotype, and here's a haplotype. We have 2 parents, one parent that gave a big D, a little c, and little e antigen, and then the other parent who must be big D negative, so they're Rh negative. They gave a little c and a little e antigen. So the offspring are going to inherit either a big D, little c, or little e, or the little d, little c, little e from the parent. They can never, ever switch around this combination, so they can't inherit a little d, a big C all of a sudden, and a little e, as this would indicate crossing over, and this does not occur in the Rh system. So there is no such thing as flip-flopping, crossing over during meiosis. It's always just going to stay as is. It will always be inherited in a linkage fashion with no crossing over. So we're still talking about Fisher-Race here, and with the exception of the little d, each allelic gene controls the presence of the respective antigen on the red blood cell. So the gene complex big D, big C, little e would cause the production of the big D antigen, the big C antigen, and the little e antigen. If the same gene complex on both paired chromosomes, so if one from each parent, one haplotype from each parent were the same, then only the D and the big C and the little e would be present on the cells. You can't add them together or anything like that. Like if they're the same, then that's what's going to be the same on the offspring's red cells. If one chromosome carried the big D, the big C, the little e, and then the other one actually carried the big D, little c, big E, this would actually cause inheritance of all of these antigens. So this would cause the inheritance of not only the big D and the big C and then the little e from the first one, we'd also be inheriting the little c and the big E from the second haplotype from the parent. So they're present. If you inherit them, they're going to be present on the red blood cell. So each antigen, except again for the little d, which is an amorph and produces a null phenotype, is recognizable by testing red cells with specific antiserum. So we can use manufactured antiserum and direct it at those antigen sites on the red cell and be able to agglutinate it and pick it up. So that, again, was Fisher-Race. The next one is the Weiner. And so, again, Weiner postulated that there was actually 2 genes. So as Fisher and Race has 3, Weiner thought there was 2 genes, one on each chromosome pair, that would control the entire expression of the Rh system. So this researcher thought that each gene produced a structure on the red cell called an agglutinogen or an antigen. An agglutinogen is a term that refers to a group of antigens or factors that are going to be agglutinated by antisera. So you can identify this agglutinogen by agglutinating each of its parts or each of its factors. So in this case, they thought that this agglutinogen, or this antigen, consisted of 8 major alleles. Again, these are the ones I've listed before. Each agglutinogen had 3 factors, or antigens, or epitopes. So they believed that there was 3 ways that you could attack this antigen. The 3 factors being the big D, the little c, and the little e. So each agglutinogen can be identified by its parts or factors if you react it with a specific antibody or antiserum. So here is your main Rh gene, your 1 gene locus that's going to be inherited and produce this agglutinogen, which again will give you 3 different factors. And then these 3 different factors are inherited, and they are then shown on the red blood cell surface, and that's how you can get 3 epitopes per this 1 agglutinogen. I'm just going to go back for a quick second. Okay, yeah. So I just wanted to show these are the 3 different parts again. And so what you need to understand is, unlike the Fisher and Race, where that had 3 separate genes at a loci, this only has 1 gene that can produce an agglutinogen. Now, the 2 theories are the basis for the 2 notations currently used for the Rh system. So immunohematologists will use a combination of both systems when we record probable genotypes. So when we're referring to the Rh blood group system, we can name the different antigens that have been inherited based off of both of these two naming systems. So you must be able to convert a Fisher-Race notation into a Wiener shorthand. So again, we'd need to recognize that if there was 3 antigens, we're talking about a Fisher-Race. You're talking about 3 different genes. And then we'd have to convert it into the Wiener format, and which is 1 gene. So given an individual's phenotype, again, we must be able to determine all the probable genotypes and write them in both different notations. So one thing I just wanted to mention here, but I'll reiterate it again in a little bit, is that R1R is the most common D-positive genotype, and little r is the most common D-negative genotype. And that doesn't make sense to you right now, but I'll explain what each of those letters mean. The big R actually means that a big D antigen was inherited, and then the little r's mean that no big D was inherited. So that's why the little r is associated with the D-negative genotype. But again, I'll talk about that in just a second. Okay, so on this next slide, you'll see a comparison of the Wiener and the Fisher-Race. Now, this might look like a lot of information, but essentially what it's showing us is that on the left side, those are all the single genes that are inherited by the Wiener pattern of inheritance, and then they produce an agglutinogen. And again, those agglutinogens have 3 different factors or 3 different epitopes that can be recognized. And based off of the inheritance of those 3 different epitopes or factors, you can actually have a shorthand way to describe that inheritance pattern, and that's what's listed on the shorthand designation. And then those shorthand designations can actually be translated into a Fisher-Race antigen system, which you'll notice has the 3 different genes there listed. So what do all these letters mean? So let's keep going. Okay, so the most important thing you need to understand when you're translating between Wiener and Fisher-Race is that the big R is the same as big D, which means that there was a big D antigen inherited. Little r indicates that there was no big D antigen. So we could use, again, the designated little d to signify that no big D was inherited. Then the number 1, which would be over here on the left side, 1, or the character little prime down here, actually denotes big C being inherited. And then the 2, or the double prime down here, indicates the letter or the antigen big E being inherited. And then if there's a 0 above or next to the big R, and if there is nothing next to the little r, then that means that there's no C, no big C, or no big E inherited. And then if there's a superscript Z next to the big R, and if there's a superscript Y next to the little r, that means that both big C and big E were inherited. Now, this is a nice little setup, but I think I'm going to show you in a different way as well. So one thing to keep in mind is that the superscripts are referring to genes, and then subscripts are referring to the agglutinogen or the complex of antigens. So if you're reading it in text format, if I said the Rh superscript 1 gene encodes for the Rh subscript agglutinogen, which is made up of these 3 antigens. So usually this can be written in shorthand, which would leave out the little h. So big D, big C, and little e is usually written as just R1. So the shorthand versions that, if I go back on this chart, they drop all of the h's here. associated with the glutinogen, and they just keep the R format. Okay, so this is kind of like what I was talking about before. Essentially, we have big R, again, which is going to signify the presence of inheriting a big D. We're going to have a little r, which is going to signify the absence of the big D, or we could put little d in its place to show that we did not inherit the big D. And then with this, I tried to just draw like a straight line down your paper so that you can associate your big R with inheriting, with having a superscript 1 next to it, a 2, and then a Z. And then the same thing for the little r. We could have a prime, a double prime, and a little y. And I know I already told you what each of those mean as far as like what's inherited 2 slides ago. But for me, it's very easy to just associate actually both the antigens with them. So when I see a 1 or a prime, I don't just think, okay, big C was inherited. I think big C little e. And then for 2, then 2 prime, even though I know it's big E, I associate it with little c big E. And then for Z and Y, I associate it with both of them. And then remember 0, and if there's nothing, then it's only the D antigen or nothing, all the small antigens being inherited. Here's another nice little chart that kind of explains it again. This is very important for you to understand. I've listed a link for some practice problems because you do need to understand how to convert between the Fisher-Race terminology and the Wiener terminology. So here's our Fisher-Race. So when you look at this, we see it's a little d. So right away that should tell us, okay, it's a little r because the big D antigen was not inherited. Then we see it's a little c and a little e. So personally, like I told you, is if I see two little antigens inherited, I automatically think either 0 or nothing. And since this is a little r, it's not going to be anything because no big C or big E antigen was inherited. For the next one, we have again still a little d, but now we have a big C and a little e, so we have a prime being inherited. Then for the next one, we have also no D antigen, but we have now a big E antigen being inherited. So now we have 2 prime. And now for the 4th one down, we see that a big C and a big E was inherited. So now we have a little y next to our little r to signify that there is no D antigen, but there's a big C and a big E antigen. And now these last 4 you'll see are all big R's because the D antigen was inherited for all of them. It's just, again, now looking at whether the big C or the big E was inherited. So in this case, the big C was inherited, so we have 1. In this case, big E was inherited, so we have 2. In this case, both big C and big E were inherited, so now we have a letter, Z. And then in this case, we have neither big C nor big E being inherited, so it's 0. So please just practice. Memorize this. It's the best that you can do to help yourself remember. Rewrite it, rewrite it, rewrite it. So those are 2 terminologies, and they describe the actual antigen terminology. They're based on genetic concepts. Now this is another system known as the Rosenfield system, and it was developed to communicate phenotypic information. So not necessarily the genetic components as far as what was inherited, but more so what are we seeing expressed. So what are we phenotypically seeing on the red cell? And it was developed because it allows for us to enter it into computer or data entry a lot easier because it's not addressing genetic information. So antigens are numbered in the order of the discovery and the recognition as belonging to the Rh system. Again, there's no genetic assumptions made. The phenotype of a given cell is expressed by the base symbol of the Rh, followed by a colon, and then a list of numbers that will represent the specific antigens. And if it's listed alone, that means that the antigen is present. So here we have Rh, semicolon. So we see that we're talking about the Rh blood group system. And I'm sorry, it's not a semicolon, it's a colon. So we have Rh colon 1. So 1, we're talking about the D antigen. So it is present. If it's listed with a dash, or like a little minus sign, then the antigen is not present. So this next example, we have the Rh blood group system, colon 1, telling me that the D antigen is present. A little -2 telling me that, okay, big C was not inherited, so we actually have little c being inherited. And then 3 doesn't have anything next to it, so I know that big E was inherited. So if it's not listed, then the antigen status was not determined. So if I had skipped 2, then that means I didn't figure out whether or not there was a little c or not, or a big C or not on the red cell antigen. But this was developed in 1962 based off of serological agglutination reactions. So the phenotypic expression of these antigens, because it adapts well to computer entry. And here's a nice comparison of all 3 systems. So again, we have our genotypes that are described using our Weiner and Fisher race system. So we're talking about inheriting genes. And then our Rosenfield, we're talking about each individual phenotypic expression of the antigen based upon whether or not there's nothing next to the number or if there's a negative. Also on the right side here, it tells you the frequencies. So I had mentioned before that R1, little r, is the most frequent genotype expression in whites. So you'll see 33% actually will have this on their red cell. They'll have a big D, they'll have a big C, they won't have a big E, they'll have a little e, and then they'll also have another little c and another little e. So 4 and 5 is then referring to the next haplotype. So we have 1, 2, 3, and then 4, 5, 6. Does that make sense? Okay. So then the International Society of Blood Transfusion, also abbreviated as ISBT, in an effort to standardize the blood group system nomenclature, they assign a 6-digit number to each blood group specificity. So the first 3 numbers represent the system, and then the remaining 3 represent the antigen specificity. The assigned number of the Rh blood group system is 004, and then the remaining 3 numbers correspond to the Rosenfield system. So for example, the ISBT number for the C antigen is 004002, and you can see here the 1 is associated with the D antigen. For recording of phenotypes, the system will follow the Rosenfield approach. So again, Rh ISBT number is 004. The number of antigens that can be found in this blood group system are between 48 and 53 antigens. So this is a very, very large blood group system. And again, sometimes it's incorrectly called rhesus, but remember the rhesus antibody is actually associated with the LW blood group system. So it will react serologically, like in our testing methods, resembling an anti-D, but it's actually not an anti-D. It's not directed at the Rh blood group system at all. It's developed at another blood group system known as LW. Another thing I wanted to mention about the expression of the Rh blood group system is that the Rh blood group system, these antigens that are inherited, are expressed on cord blood cells. So they're expressed very early on in a fetus's development, and then they're also expressed obviously on adult red blood cells. There is no soluble form which is the case for the ABO blood group system, so this is different. And then it does not appear to be expressed on any other tissues. So it's very, very erythroid or red blood cell specific. Now, the antigen structure of the actual Rh system is that it is, they are proteins, so they are made up of 416 amino acids that actually go and traverse the red blood cell membrane about 12 times, and they display short loops of amino acids on the exterior. The short exterior loops of the amino acids are the ones that make up the Rh blood group antigens. So here's a nice picture, so you can see that here is our Rh protein that's traversing the cell membrane 12 times, and it is those short loops at the top that's going to be the sites for our antigens. And this is the correct order, because you'll see that big C and little c, they do vary in their amino acid sequence. So big C is actually known as a serine, and then little c is known as a proline. Again, that's what is going to determine which antigen is there. It's going to be determined based off of which genetic amino acid sequence was inherited, but they're both found at this location, 103. And then the big E and the little e are both found at 226. That location, again, the inheritance of each of those is going to be determined by your genes, and it's going to be determined whether or not a proline was inherited or an alanine was inherited. Okay, so we need to remember again, I've been talking about phenotypes and genotypes. So the phenotype is the result of the reaction between the red cells and the antisera. It's what we see serologically. The genotype is the genetic makeup, which can be predicted by looking at the phenotype or the reactions that we're having, with our methods. And then we consider the race of the individual to determine what genotype they might have. So only family studies can truly determine the true genotype, so we would have to look at an entire family in order to determine what the actual genotype is. But we can look at the phenotype and then, based upon the race, determine a probable genotype for that patient. So there are 5 reagent antiseras available. There is anti-big D, anti-big C, anti-little c, anti-big E, and anti-little e. Only anti-big D is required for routine testing, and you will see that because it's included in our forward type of our blood typing procedure. The other typing sera are used for typing red blood cells, whether it's to resolve an antibody problem, to antigen type, or to conduct family studies. So what we can do with these reagent antiseras is look for agglutination reactions. Again, if there's agglutination, we know it's positive, and if there's no agglutination, we know it's negative. So that's going to represent our phenotype. That's what we're seeing serologically. There is no anti-little d again, since that truly is not a real gene, and since little d is an amorph. But we can use statistical probability to determine the most probable genotype based off of the phenotype that we're seeing in these agglutination patterns. So we use what's called phenotyping, or Rh phenotyping, with those 5 reagent antisera in these 4 different situations. We use it in parentage testing. We use it to predict hemolytic disease of the fetus and newborn. We use these reagents to confirm an Rh antibody specificity. We can use these reagents to locate compatible blood for recipients who have these Rh antibodies, because if they have an Rh antibody, again, we want to make sure we're giving blood that's negative. for that specific antigen, and we would use those reagents. And we'll get into that during in-person class in January. But essentially what you're going to do is you mix your red blood cells with the Rh antisera, and we look for agglutination patterns. The agglutination, again, will indicate the presence of the antigen on the cell, and it helps us to determine the phenotype, or the actual expression of the gene. We can use published frequencies and subject information to determine their genotype. Now, molecular testing is becoming a lot more popular, and there's actually a lot of seminars and audio conferences that are mostly interested with this molecular testing of the Rh blood group system, since it is so immunogenic. It's very interesting to sit on one of these seminars or audio conferences and listen to the pros and cons of molecularly testing. Again, cost always comes into play. However, molecular testing is really, really good because we cannot use antisera on recently transfused individuals. So molecular testing can be used to differentiate the Rh blood group system on these individuals. And these individuals usually are like sickle cell patients who are constantly getting transfused because of their condition. They're usually very, very anemic. And they are actually prone, due to the large amount of blood transfusions they receive, they're prone to developing antibodies at one of the 50 antigen sites of the Rh blood group system. Because there are so many, they actually start to develop these variant antibody productions. So it's best if we can molecularly test them, even if they've been recently transfused, so we can determine what is the most compatible blood that we can give them for them not to have an immune response. So again, we can use molecular testing on recently transfused individuals. Antisera is not available for some of those rare variant Rh antigens, as well as other blood group systems. So molecular testing would be really, really good in testing for those genes as well. We can also use it to test D-zygosity. So that's talking in regards to the variant types of the D antigen. So we'll get more into the structure about the D antigen, but essentially we're going to talk about like partial Ds and weak Ds, and these antigens are very, very prone to being manipulated so that some of their parts or all of their parts may not be inherited. So these patients whose parts of their full D antigen are not fully expressed are more prone to developing antibodies. So it's important for us to realize that they are a zygote and that they do have this variant inherited form in order for us to, again, know what type of blood we should be giving them. We can also use molecular testing for fetal genotyping for D, and you can do this on fetal DNA that's present in the maternal plasma. So there are techniques that physicians can use known as cortocentesis, as well as chronic villus sampling, where they actually have to do a, a very, very intense surgery on the mom in order to go in and obtain these samples from the fetus. However, this is a great way with this molecular testing, just by obtaining a mom sample, not having to do this very, very complex surgery. Molecular testing also is great with monoclonal reagents from different manufacturers, which are going to react differently with variant D antigens. We have seen in my lab especially where we have two different types of manufactured anti-D reagent, and the monoclonal reagents are not going to react with some patients' D antigens, whereas a polyclonal blend might. And we can eliminate the need of having to test with both different types of reagents if we just molecularly test everybody. So that's another pro as well for beginning to do molecular testing. Typing sera continue to be the gold standard, but eventually I really do believe, as long as the funding comes in, that molecular testing is going to be the way of the future. And I talked about determining genotypes by looking at phenotypes. Well, the one thing you always want to do is determine the race of the patient. Because as you can see here, Caucasians and Blacks, they do have varying genotypes. So in the Caucasian population, you'll see that the R1 genotype is the most common. So we're talking about a big D and a big C being inherited, and then a little E being inherited. And then the Blacks, we're talking about R superscript 0. So we're talking about a big D being inherited, a little C, and a little E being inherited. There's a great table, 5-4, in your book that explains the highest frequencies in the population based off of each of these races. But this is something that we could use to determine a patient's genotype if we are phenotypically antigen typing them using our test methodology. So again, refer to your textbook. Genotypes are listed as presumptive or most probable because we can't really be 100% sure. They vary again with different racial groups. So here again is a nice chart that shows Caucasians 40% are R1, followed by the 38%, which is little r. So most inherited haplotypes, like from one mom to one parent to another parent, would be big D, big C, little e / DCE. So that's usually the most common in Caucasians, whereas with Blacks, again, we're looking at R superscript little 0, and then the next most common is the little r. So the most frequent genotypes in Caucasians, again, is R superscript 1, little r, and then in Blacks, it's R superscript 0, little r. And now, based upon which genotype you are and how you phenotypically express these antigens is going to determine the amount of D antigens on your red cells. So the D antigen concentration does vary with the antigens that are inherited by the gene. So you'll see that the most D antigens are actually on what's known as a D deletion phenotype. So that's that first one up here. That's when you have a person who only develops a D and does not develop any other antigens, but that's very, very rare. The next most prevalent D antigen we see is on an R2 patient. So again, that's the most D. R1 is next, followed by R1R, R0R, and then so on and so forth. So it's important to recognize that each of these different genotypes are going to have different expressions of D antigens. Specifically, when we start to look at our screening cells that we're going to use to look for antibodies, we're going to be using R2 cells because we want to have a high expression of the D antigen. And that should probably be our screening cell number 2 when we get to it. But you'll see, based off of the different genotypes, it's going to determine how many antigens are on each of those cells. So again, these proteins, except for the ABO, the Rh antigens are more numerous than any other blood group system. And they're only found in our red blood cells, as I mentioned previously. And this is explaining the immunogenicity. And again, immunogenicity is the ability of the antigen to stimulate an immune response. So what we find is that D, if a person is exposed to a foreign D, that's the most immunogenic. Patients will develop an anti-D very, very rapidly, followed by little c, then big E, then big C, and then little e. So most red cells can be typed for the D antigen directly with anti-D reagents. However, sometimes the D antigen is weakly expressed on the red cell, so its detection actually requires the use of another test. It's known as an indirect antiglobulin test. So this weak D is a weak form of the D antigen that's going to require the indirect antiglobulin test for its detection. So Red blood cells, normally you can put them in a tube with the reagent Antisera and spin it in your centrifuge and, check for agglutination. With these weaker expressions, we have to allow for more time. We have to allow for those two processes, the sensitization process of agglutination and the lattice formation of the agglutination to have an effect with each other. So what we actually wind up doing is called the indirect antiglobulin phase. So we incubate cells, the patient's red cells, with the anti-D reagent, and we incubate this at 37 degrees Celsius. Now this is like the temperature of our body. However, we do things at room temperature. So what we have next to our stations are known as heat blocks. So we would put the tube in this heat box, and then we allow about like 15 minutes for the coating of the D antigens to occur if it's present. So we have this coating of the anti-D reagent on these weaker expressions of the D antigen, if they're there. Then what we do is we wash the cells 3 times with saline, and then we add another reagent known as anti-human globulin reagent. And the anti-human globulin reagent is going to bind to any anti-D that's coating the cells if the original D antigen was present, and we'll look for agglutination. If it's negative, then the individual never had D antigens to begin with, so they're D negative. And then if it's positive, then the individual must just be a weaker expression, and they are still D positive. Now you might be wondering, like, well, what makes one person a weaker expression of D compared to others? There's 3 different ways that you could have a weaker D expression. The first way is a genetic way. And genetic meaning you might just have a different gene that's going to code for a weaker expression of the D antigen. It could be a position effect. So sometimes you have weaker expressions of D based upon where your certain genes are inherited in relation to the other genes. So if they're on the same chromosome, then they're usually in a cis position. But if they're on opposite, they could be in a trans position. You could also have mosaic forms. But essentially, these 3 different mechanisms result in differences from the normal D expression. So you have quantitative differences, which would be your genetic or your inherited genes or your position effects. So you actually have a decrease in the number of D antigens. It's a quantitative difference. Or you can have a qualitative difference, such as the case with the mosaic form. And that's usually because you have the D antigen, but there's something slightly different with the actual appearance of the antigen. And I'm about to get into it a little bit further, so it'll make a little bit more sense. So genetic, again, is quantitative. We're talking about the gene expression resulting in fewer or more antigen sites. So you have the R0, which is common in blacks. And that's actually a very, very decreased expression of the D antigen. And then the R1 and the R2 in Caucasians is also a weakened expression of the antigen. Then you have a position effect. Like I told you, it depends how the genes are inherited on the chromosomes. This is also a quantitative difference because we're affecting the number of antigen sites on the red cell. So what this is talking about is having the inheritance of the C gene in a transposition to the D gene. So we're talking about inheriting 2 haplotypes, one from each parent, and one parent is inheriting the big C. So you'll see here they're pairing an R prime, or the big C, little d, little e, with a R1 or R0. So you'll notice that the D antigen is technically on the one haplotype, and then your big C is on the other haplotype. that's putting them in a trans position, and that can result in a weakened expression of your D. And then finally, you can have a qualitative difference in partial Ds, which it's also known as a D mosaic or a D variant. That's older terminology, but you might see that. But essentially, this is where you have the D antigen is made up of different epitopes, and one or more of the epitopes is actually missing. So it's a qualitative difference because we're not talking about difference in numbers of antigen sites, we're talking about difference in the actual structure. So I'm just reiterating again each of the 3 different mechanisms. And here's our genetic difference where we have normal amounts of D antigen and then we have less amount of D antigen because of a gene inheritance. I talked about the position effect where the C and the D arrangement, because they're on opposite haplotypes, they actually cause what's known as steric hindrance, and it results in a weakened or suppression of the D expression. So right here, we're talking about the D is on the one haplotype and the C is on the other haplotype. So if you put them into, like, you could even draw a chromosome right now, and you would see that they're on opposite chromosomes, so they're in a trans position, and this causes steric hindrance. Whereas over here, it's in a cis position because on one chromosome, you would have them both inherited. So you would not have a weakened expression. And here's actually the nice picture that I wanted you to draw. So we have our cis, and then we have our trans. And our partial D, like I was talking about, here's a nice picture that shows the difference. Down here, you'll see this is a normal D antigen site with different epitopes or binding sites. And then here's your red blood cell, but all of a sudden, uh-oh, we're missing a portion. And this missing portion can cause it to be more difficult for us to pick up the expression of the D antigen. So if a patient is transfused, so let's say we do try and type this person who has a weakened partial D, and we type them as being Rh negative. So we didn't see the D antigen, and they go to donate blood, and they're actually given to a patient who is truly Rh negative. There still are these other remaining parts here that make it technically D positive. So a person who's Rh negative and is exposed to a partial D is capable of making an anti-D alloantibody, specifically to the part that it's missing. So this person can also make an alloantibody to this specific part that it's missing. So not only can they harm recipients as a donor, but they can also themselves make an allo-anti-D to that specific part that they're missing. So these are the partial D situations that are a little bit more complicated, and that's why a lot of immunohematologists want to do molecular typing, because if we knew in the beginning that this person was a partial D, and we give them blood, we could determine exactly what type of blood they need so that they don't produce this anti-D alloantibody. And now out of all the 3 different varying expressions of the D antigen, this is the only special type that can produce the anti-D alloantibody. And again, an alloantibody is an antibody that's produced with specificity other than itself, so it's directed at another foreign antigen. And here's what I was talking about, the significance of weak Ds. So for donors, if you're a weak D person, we're still going to label your unit as D positive, and that again is because if we were to label it as D negative and give it to a recipient who is truly D negative, they could have severe hemolytic transfusion reactions and develop an anti-D. So we're always going to label donors as D positive. And then in patients, if they're weak D, especially known to that partial D that I just discussed in the previous slide, which can make the antibody, we actually will note that they're weak D positive, but we'll transfuse them with D negative blood so that they don't produce that alloanti-D. So we do weak D testing on donors for that reason. And then we do weak D testing on some patients, but it's not required in certain situations. So we really only do weak D testing in my blood bank on pregnant women, but all adults otherwise, we do not do weak D testing. Okay, you might want to take a breather now because what we talked about up to that point was the D antigen, and we talked about the other 4 Rh antigens, your big C, little c, big E, and little e. And what we're about to get into right now is what's known as compound antigens. So I told you that the Rh blood group system is very unique in that it's made up of more than 50 antigen sites. Well, some of them are very, very complex, so depending upon which antigens are inherited, you can actually have other antigens created. Okay, so compound antigens are epitopes which occur due to the presence of 2 Rh genes found on the same chromosome, so they're in the cis position. These gene products include not only products of a single gene, but also a combined gene that's also antigenic. So examples of compound antigens in the blood group system are as follows. You have the F antigen. So there's a nice chart in your book, and it lays it out pretty nicely. But the F antigen is going to be found whenever you have a little c and a little e in the cis position, such as the example of little r that's right here. You have D, little c, little e, and a D, little c, little e. So this gene actually codes for the F antigen because the little c and the little e are in the cis position. The F antigen will not be present in the trans position. So this is actually the Rh6 blood group system. I told you there's many different antigens, so this is just one. The next one is the Rh little 1 or the big C little e antigen. This is also known as Rh7. And it occurs whenever there's a big C and a little e in the cis position. So what's important about these compound antigens is that if they're inherited in this pattern and the person is positive for that compound antigen, that's great. What happens is when a person is not this genotype and does not inherit these antigens in this cis fashion, they're prone to developing antibodies to these specific blood group antigens. So antibodies are rarely encountered, but if an individual had an anti-F, it would only react with the F positive cells and then not cells that were positive for little c or little e. So it's possible for these people to develop antibodies, and that's exactly what it would look like. It would only react with cells that were F positive. F cells are clearly marked on antigram of screen and panel cells. So when we get out into the lab, manufacturers make sure that we know that F cells are present. And so here's a nice chart as well. So you can see that the ISBT symbol associated with each of these antigens on the same haplotype on the cis position. And then the other common names, we have F, we have Rh little I, and then we have the Rh22 and the Rh27. So it's just important to recognize that if these antigens are inherited in a cis position, they are a blood group antigen as well. So here's a nice little chromosomal picture for you as well, so you could see that if there's a big C and little e inherited, that's Rh little I. If there's a little c and little e inherited in the cis position, it's an F. Little c big E is your Rh27, and then your big C big E is your Rh22. So then you also have a G antigen. So genes that code for both big D and big C antigen are actually negative for the G antigen. So genes that code for big C or big D separately are also coding for big G. An anti-G mimics an anti-C and an anti-D. So we actually cannot separate out an anti-G from an anti-C and anti-D unless we use like adsorption and elution studies, which we're not going to talk about right now. But generally, it's important for us to recognize whether or not it's an anti-G antibody or an anti-C or anti-D antibody. Usually, in the cases with like hemolytic disease of the newborn. So again, the G antigen is expressed whenever there is a D or a C antigen. There's only in cells that are negative for both D and C is when you'll have a cell negative for the G antigen. So then there's unusual phenotypes that can result in the Rh blood group system. They're rarely encountered in routine blood bank testing. However, we have to be aware of them and we have to understand their inheritance patterns and the cell characteristics. So we talked about null phenotypes. So the null phenotype, again, was the absence of a particular blood group system from the red blood cell membrane. And we said that that could occur because of the RAG co-expressor not being inherited along with the Rh blood group system. But here's another situation where we'd also have a null phenotype, and this is known as a D deletion. So this is a very, very rare Rh phenotype that demonstrates no reactions when the red cells are tested with any of the 5 reagent antisera that we talked about, the anti-big E, the anti-little e, the anti-big C, and the anti-little c. Basically, genetic material has been deleted or rendered non-functional. The red cells that lack the big C, little c, and the big E, little e antigens, they actually demonstrate very, very strong reactions with D antigen activity. And what that means is if you go back a few slides, you'll see that this D deletion phenotype is actually going to have the highest concentration of D antigen sites. So if a person who was a D deletion needed a transfusion, we'd only be able to give them other D deletion red cells, because otherwise they would definitely produce antibodies to these single or separate specificities. So if you ever see D dash dash or dash D dash, that's talking about this D deletion phenotype. So that was the first type of null phenotype, D deletion. The next one is the Rh null phenotype. And so this is for patients whose red cells have no Rh antigen sites. So it's usually written as like dash 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 slash dash 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 to show that they've inherited 2 haplotypes, one from each parent with nothing associated. So the lack of antigens causes the red cell membrane to kind of appear abnormal, and it leads to stomatocytosis, which is like a rounding of your red cells. So normally your red cells are like a biconcave shape. Well, stomatocytes are going to be very, very spherical. It can lead to hemolytic anemia. And there are 2 Rh null phenotypes. The regulator type, where it's gene inherited. So you might have inherited the actual Rh gene, but it's not expressed. And that's usually because of, like I was saying, the regulator gene RAG that should have been inherited on chromosome 6. might not have been inherited, and because it's present is essential for the expression of the Rh system. This regulator type is causing your Rh gene, even though it was inherited, not to be expressed. And then there's the amorph type, so cells that are totally Rh negative. It's because the RHD gene is absent, so they're not going to have any expression whatsoever. And these patients are actually capable of making what's known as an Rh29 antibody. It's a total Rh antibody, and if you detect an anti-Rh29, you need to give this patient only Rh null cells for transfusion. So normally, whenever a person is Rh null, you would usually look for like donations from siblings or autologous donations, and that means like donating for yourself. And there was a man, I read an article, I wonder if I can get it up for you and link it on here. He was in Britain, and he is the Rh null phenotype, which is like really, really, really rare. And he had to donate his own blood and have it frozen for him so that he would be able to receive blood if he needed it in the future. So they were able to freeze it for him. So he has to go donate like every 10 years, I believe. Then this is the other blood group system that I talked about. I just wanted to mention it again so you like can make that distinction between a real Rh antibody and a rhesus antibody. So it was discovered at the same time, but LW is detected on cells of like rhesus monkeys as well as human red blood cells. It's found in the same proportion as the D antigen, and even though it was thought to be the same antigen, they did discover differences. So rare individuals that lack the LW can have also normal Rh antigens. However, these patients that lack the LW antigen can form an allo anti-LW, and the anti-LW will resemble an anti-D. However, when we actually start to go and do serological testing, what we're going to find is that the anti-LW is going to react more strongly with D positive cells than D negative cells. And we need to keep in mind that when D positive individuals appear to have an anti-D, they might actually have this anti-LW. Another antigen, a variant Rh antigen that I wanted to mention is the CW. You will see this on a panel. It's a very, very low frequency antigen, so not a lot of people in the population have this antigen. And when you actually get to check it out on the panels, you're going to see that out of like 16 or 20 cells, there's only going to be about 1 or 2 cells that are actually positive for this antigen. Because it's only found in about 1 to 2% of the whites, and it's very, very rare in blacks. But most individuals who are positive for this antigen are also positive for the big C antigen. So generally, if a person does develop an antibody to this antigen, it's because they lack the big C antigen as well. And antibodies to these antigens can be naturally occurring, and they actually are important because they can play a role in hemolytic disease of the fetal newborn, and they can cause hemolytic transfusion reactions. So those are two key things. I don't know if I've stressed that enough, that it's going to determine the significance of antibodies. Is it capable of causing hemolytic transfusion reaction, and is it capable of harming a fetus and newborn? So all the Rh antibodies, except for the rare examples of your anti-CW, which may be naturally occurring, most occur from immunization due to transfusion or pregnancy. And right here, it's associated with HTR and HDFN. So that's why all Rh antibodies are important. They're very significant. The key characteristics about Rh antibodies is that they're IgG primarily. They might have a minor IgM component to them. So they should not be reacting at immediate spin or saline suspended cells, because that's happening at a room temperature phase. So really what we'll see is these Rh antibodies, when we get to testing, we'll be picking them up at 37 degrees Celsius because that's like our body temperature. And then we'll definitely be picking them up at the last phase, which is known as the IAT or AHG phase. And then these Rh antibodies are enhanced if we were to use special enzyme treated cells. They're actually enhanced. So again, the order of immunogenicity or the ability of them to cause an immune response is D, little c, big C, and little e. Another thing is that they do not bind complement, and then they actually will cause extravascular hemolysis. Anti-E is most frequently encountered antibody, followed by anti-little c. Anti-big C is rare. Anti-little e is rarely encountered. Only about 2% of the population is negative for this. So we rarely see it as an antibody. The times we do see it is generally in like the sickle cell population or patients who have received multiple transfusions. Detectable antibody persists for many years and sometimes for life. So once you identify a person as having an Rh antibody, we must always give them blood that's negative for that antibody, even if it's not currently demonstrating today. So maybe we detected an antibody like 2 years ago, but it stays with them and their patient chart for life. So if I were to go into the computer and I was getting ready to set blood up for somebody and I saw 2 years ago we had agglutination and it proved to be an Rh antibody, I can't go, oh, well, it's not demonstrating today, I'm just going to give them any blood that I deem necessary. No, that's not possible. You can still kill somebody because they can have that secondary amnestic response, so you must still give Rh compatible blood. Anti-D can react more strongly with the R2 cells, and that's because we talked about them having a higher concentration of the D antigen sites. And then you can have some antibodies that occur together. We talked about the G antigen, and of course you can have an anti-G develop in a person who doesn't have a C or D antigen. So in that case, you could actually have two antibodies developed. You could have an anti-D and you can have an anti-G. You can also have that anti-little f antibody develop, and oftentimes we see it in combination with an anti-little c. What's most important is that R1 people can make an anti-E frequently, and then they frequently also make an anti-little c. Because what is R1? It's big D, big C, little e, / big D, big C, little e. So they do not have the big E, and then they also do not have a little c. They have a big C. So that's why people who are R1 can make anti-big E, and then they frequently make an anti-little c as well. So patients who have this anti-big E should also be phenotyped for the C antigen as well. And if a patient appears to be R1, they should be transfused with R1 blood. If not, then they have that possibility of creating an anti-little c. So there's 4 different types. We talked about how there's a reagent antisera that we can use. Well, specifically, there's 4 different types of anti-D reagents. There's high protein, there's IgM, chemically modified, and a monoclonal source. So the high protein reagent is faster, it has increased frequency of false positives, and it requires the use of an Rh control tube, and it can convert to weak D testing. So it has its pros and its cons. It's faster, and you can use it to convert to your weak D testing if you need it to be, so you wouldn't need to have 2 separate bottles, you could just use the one. However, the problem with it is that it has a frequency of false positives, and then it does require an extra step. You'd have to throw in a control tube, so you kind of have to weigh your pros and cons. Then you have IgM, which is a low protein, and it's a saline reacting anti-D reagent. So This lower protein actually will allow there to be fewer false positives, so that's a positive. The only issue is that it's kind of a longer incubation time, and then it can't be converted to weak D testing. Then you have a chemically modified anti-D reagent, where this is like a relaxed form of the IgG anti-D, and it's in a low protein medium, so you do have a few false positives. You do need to have a saline control performed along with it, and then you are able to convert this to weak D testing. So that sounds good. And then there's a monoclonal source, which is low protein, so it'll be fewer false positives. And then it's usually you have a blend of different monoclonal antibodies. So specifically in my blood bank, we use monoclonal sources, and they're good, except for the fact that we talked about we know monoclonal antibodies are only directed at one specific sort of epitope. So if for some reason a person was like a partial D or a D variant, we might not be picking up their cells as being Rh positive, because we're not using like a polyclonal blend. We're not using a chemically modified or high protein blend. But we use monoclonal because it's a lower protein, and we do like that it only detects like the one specific epitope. So you need to know the preparation, the use, and the advantages of each of these. When you get out to the blood banks, you might find that they're using high protein. I believe that a majority today are using the monoclonal sources, but you need to understand the differences. You need to know when you might need a control tube, and you need to also understand, like, is there a possibility of there being a false positive? Yes. So the high protein anti-D reagent is potentiated with high protein and other macromolecules to ensure agglutination. We said it might cause false positives, so you always need to have a control, so that your control should be negative in order for you to ensure that the positive reactivity you're getting is truly positive, and it's not due to a false positive because of like abnormal serum proteins or other antibodies to the additive of the actual reagent. Here's your IgM anti-D. I'm just going to quickly go over them because I just kind of discussed them, but make sure you look at them and you know the difference. And here's the monoclonal one. You'll see it says it's the most frequently utilized reagent. And the reason being is because we'll react at immediate spin, but then we also will use it for the weak D test. And the only time we really need to do it at control tube is when we have an AB positive person. Precautions for Rh typing is you must follow manufacturer's instructions. So with each of those reagents that we're going to be using, there's a specific set of guidelines that have been given to us in a package insert. We cannot use those reagents for the indirect antiglobulin testing for our weak D patients unless it explicitly is instructing us to. And then, of course, you're always going to be running controls with each of these reagents. So you'd be performing QC daily if you're going to be running that anti-D. In my blood bank, the first person who needs to use that reagent antisera for the day will QC it. And as long as we continue to use that same lot of anti-D, then we do not need to QC it again for the remainder of that day. Sources of errors. So false positives can occur. We need to remember that there can just be spontaneous agglutination. You could have contaminated reagents. You might be using the wrong typing sera. There's something known as autoagglutinins or abnormal serum proteins that tend to coat red blood cells, and they can make it look positive when it really isn't. So using antisera in a test method other than required by the manufacturer, if you're not following the exact directions outlined by the manufacturer, you could develop a false positive. False negative. So let's say you're going to use that reagent, you're trying to see if there's the antigen present on the red cell. Well, it might be falsely negative because you used the wrong antiserum. Maybe you failed to add the antiserum to the actual tube. You might have thought you added it, but you didn't. Maybe you had the incorrect cell suspension, so you had too heavy a suspension. You had the incorrect cell to serum ratio, so generally it's just like 1 drop of your antisera to 1 drop of your cells that you're antigen typing, but maybe you did that wrong. Perhaps you're shaking the tube too hard, so you're breaking up the agglutinates. So that's something we're gonna practice when I have you in lab. You don't want to shake it too hard, because then you're gonna keep shaking all the agglutination away, and you'll call it a negative when really it was positive. Maybe the reagent deteriorated and now it's not working any longer. Perhaps there was a failure of the antisera to react with the variant antigens. So as I told you, you can have different variant antigens, so perhaps the specific monoclonal anti-D is not directed at the correct epitope. Antisera in which the antibody is directed against a compound antigen, often problems with anti-C. So you might get a false negative because it's not picking up the correct antigen. In summary, the Rh system is second to the ABO in transfusion medicine, mainly because it's so large and it's very, very immunogenic. The correct interpretation of D is essential to prevent immunization of D negative, which may result in hemolytic disease of the fetus newborn, also hemolytic transfusion reactions. It is the most polymorphic of all blood group systems, meaning that there are many, many different forms, as you probably realized after this lecture, as well as from reading. And of the 5 antigens, only D testing is required. So that's why we only really test the D in our forward type. Only if a person were to develop an antibody will we go ahead and start to use the other antisera for the other antigens. And as the Rh system continues to grow, the last decade has led to an abundance of information detailing genetic diversity of the Rh locus. It has exceeded all estimates predicted by serology. So there's well over 100 D genes and more than 50 different CE genes that have been documented, and new alleles are constantly being discovered. And here's my references. Thank you, guys. I'll see you at class.